Hello everyone. Today let us discuss the tumors of brain. Before knowing about the tumors, let us learn about the brain cells. They are of two types. One is neuronal cells, that is neurons, and glial cells. Glial cells are nothing but supporting cells, and these glial cells are further divided into macroglial cells and microglial cells. Microglial cells are nothing but the macrophages in a modified form. These macroglial cells are of three types: astrocytes, oligodendrocytes, ependymal cells. Now let us learn the grading of the tumors before knowing the types of tumors. Grading. There are four types of grades: 1, 2, 3, and 4. These four types of grades are determined by four factors, that is atypia, mitosis, endothelial proliferation, and necrosis. Now I am labeling it as number 1, 2, 3 and 4 for our convenience. When it comes to grades, in atypia there is no in grade 1 there is no atypia, no mitosis, no endothelial proliferation and no necrosis. So there is nothing. Everything is negative, and grade 1 is completely benign tumor. In grade 2 there is atypia, number 1. In grade 3 there is number 1 and number 2, that is atypia and mitosis can be seen. Coming to the grade 4, it has grade 1, grade 2, but any of these 3 and 4 can be present. Either grade 3 or grade 4 can be present. Now we understood the grading of the brain tumors, right? Now let us enter into the topics. Firstly, we know that the microglial cells, that is macrophages, they does not give rise to any type of cancers. So they are of no value in this discussion. Now let us discuss about the astrocytes. Astrocytes can give rise to all four types of grades from grade 1 to grade 4. It can be completely benign or malignant. The type 1 or benign type of astrocytoma, the grade 1 or benign type of astrocytoma, it has another name. The another name of the grade 1 astrocytoma is juvenile pilocytic astrocytoma or pilocytic astrocytoma. Okay. Whereas the grade 4 of astrocytoma is called as glioblastoma multiforme. Remember, the juvenile pilocytic astrocytoma and the glioblastoma multiforme, both of these are astrocytic tumors. There are grade 2 and grade 3 also. The grade 2 is called atypical type, and grade 3 is called as anaplastic. That is another discussion. The second grade tumor is called as diffuse astrocytoma. That is diffuse astrocytoma. Whereas the third type is called as anaplastic astrocytoma. Now we are sorted with the names of astrocyte tumors. Number 1 is, let us revise again. Number 1, grade 1 astrocytoma. Grade 4, glioblastoma multiforme. Grade 2, diffuse astrocytoma. Grade 3, anaplastic astrocytoma. Okay. Juvenile pilocytic astrocytoma. Just remember only few things about this, very important. In cerebellum, due to BRAF mutation, cyst is seen in less than 5-year-old child. It has good prognosis because it is grade 1, right? And apart from this, microscopically there is one important thing called Rosenthal fibers. Now here I will show you where are these Rosenthal fibers. These pink colored things are called Rosenthal fibers. Apart from this Rosenthal fibers, there are cystic microcysts present in this tumor. See here, you can see the microcyst. So microcyst and Rosenthal fibers is the characteristic feature of juvenile pilocytic astrocytoma. I am writing it here. Rosenthal fibers and microcyst. This is all about juvenile pilocytic astrocytoma. Now let us go into the grade 4 tumor of the astrocytes, 4th grade. That is nothing but glioblastoma, right? Glioblastoma multiforme. Note one thing, that wherever you find blastoma word, it may be either glioblastoma, neuroblastoma, meningoblastoma, whatever it is, blastoma refers to grade 4 tumor. Blastoma refers to grade 4 tumor. It has bad prognosis. Now let us learn some points about the glioblastoma. Glioblastoma, late metastasis. In early stage of the disease, it does not show any metastasis. It shows very lately metastasis. Despite being the grade 4, it is aggressive. Okay, now glioblastoma. Glioblastoma etiologically, because of IDH, isocitrate dehydrogenase enzyme defect is the cause of glioblastoma. There are two types of IDH enzymes which cause glioblastoma. The wild type of IDH defect and mutant type of IDH. Wild type of IDH has bad prognosis, but mutant type of IDH has good prognosis, somewhat better than wild IDH. Coming to the percentage, the wild IDH has 90% of the glioblastomas are caused by wild IDH. but the mutant IDH, which has better prognosis, is around 10%. Now, microscopically speaking, there are two main important features, that is pseudo-palisading of the tumor cells and glomeruloid bodies. We all know that glioblastoma is type grade 4 astrocytoma. Grade 4 has mainly, we will say, there are two features, that is either necrosis or the endothelial proliferation. Here, surprisingly, in glioblastoma, you can notice the I am marking as a yellow color here. This is the necrosis, and this blue color thing which I am marking, this is endothelial proliferation. See here, in the necrosis region, surrounding this necrosis, I am highlighting it with blue color, there is arrangement of tumor cells along the periphery of this necrosis area. This is called as palisading. See here, I highlighted beautifully. Central thing is yellow color, that is necrosis, and the surrounding blue color is the palisading of the tumor cells. Glioblastoma, the fourth grade, it has when it has necrosis, palisading of the tumor cells around that necrosis. So this is one feature. And coming to the second feature, that is endothelial proliferation. Here I am highlighting with the green color. The red color thing which is inside here is called the blood vessel. The blood vessel endothelium gets proliferated. In proliferation, it forms a typical structure called glomeruloid body. Glomeruloid body is nothing but the blood vessel surrounded by tumor cells. So here we understood about glomeruloid body and pseudo-palisading of the tumor cells. Now let us see the macroscopic picture of glioblastoma. Here we can see the butterfly glioma, that is symmetrically on both cerebral hemispheres. We can see the butterfly glioma. It has the serpiginous necrosis. We showed na, it is in form of snake. Serpentine or serpiginous necrosis, that is one thing. Butterfly glioma, serpentine necrosis. Okay, let us revise the glioblastoma once again. 90% due to wild type of IDH, which has bad prognosis. Only 10% due to mutant IDH, this has better prognosis. But overall, glioblastoma being the grade 4 astrocytic tumor, it has bad prognosis. Okay. Microscopically speaking, the pseudo-palisading and glomeruloid body, these two are very important, and butterfly glioma and serpentine necrosis are the other two features. Okay, now we completed astrocytomas, that is astrocytic tumors, grade 1 juvenile pilocytic astrocytoma and grade 4 glioblastoma multiforme. We are wrapped up the astrocytomas. We are discussing the microglial tumors, that is the macroglial cells. Microglia does not give rise to tumors. The macroglial cells are of three types. Astrocytomas we completed. Next, we are left with the oligodendromas and ependymomas. Oligodendroma and ependymoma both are grade 2 tumors. They are not grade 1 or grade 4. Oligodendroma and ependymoma are only grade 2 tumor. Coming to the treatment portion, we did not discuss in the astrocytomas. For grade 1 tumors, surgical excision is enough. That is for pilocytic astrocytoma, surgical excision is enough. But for the grade 4, that is glioblastoma or any blastoma for that case, surgery, chemo and radio should be given. combined. Let us learn about oligodendroglioma. Here, the oligodendroglioma, we actually know that it is grade 2. The reason is just like glioblastoma, it also caused due to IDH mutation. There is one more genetic factor, that is co-deletion of chromosome 1p and 19q. 1p and 19q get co-deleted. That means 1p and 19q are deleted together. It has good prognosis. When this genetic factor is present, oligodendroglioma shows good prognosis. Repeating once again, IDH mutation, chromosome 1p and 19q co-deletion. This co-deletion shows good prognosis. Clinically speaking, in the frontal lobe, calcification occurring tumor, that is oligodendroglioma. Just remember that patient presents with tumor mass in the frontal lobe with calcification, that is oligodendroglioma most probably. Okay, coming to microscopically speaking, here you can see the image, fried egg appearance. I will show you. Here you can see centrally there is nucleus. I am marking it with yellow color, and peripherally you have peripherally there is a halo. I am marking it with white color. When you see this, it looks just like a fried egg. The yellow in the center and white in the periphery. Just like that, yellow in the center, white in the periphery. You can see many fried egg appearances in this tumor of oligodendroglioma. Fried egg appearance, oligodendroglioma. There is chicken wiring of the blood vessels. Here you can see there is chicken wiring of the blood vessels. This chicken wiring of the blood vessels. Apart from that, it has a good response to chemotherapy because it is grade 2, not grade 4. Okay, so I am repeating again. Oligodendroglioma, IDH mutation, co-deletion of chromosome 1p and 19q, good prognosis. Fried egg colonies, fried egg appearance, chicken wiring of the blood vessels. Next, frontal lobe tumor with calcification. This is all about the oligodendroglioma. Now, let us have a list of the commons. First, we will talk about the isocitrate dehydrogenase mutation. Here you can see 3 tumors will show isocitrate dehydrogenase mutation, that is glioblastoma, the wild type and the mutant type we studied previously. Then oligodendroglioma, right now we studied. Next, enchondroma. These 3 tumors show IDH mutation. Fried egg appearance. It is seen in oligodendroglioma, hairy cell leukemia, seminoma, dysgerminoma, seminoma in the testes, dysgerminoma in the ovaries. Coming to the microbiology portion, in mycoplasma and malassezia furfur, on culture media shows the fried egg colonies. Okay, let me repeat again. Oligodendroma, hairy cell leukemia, seminoma, dysgerminoma are the pathological aspects. Microbiologically, mycoplasma and malassezia furfur. Hope I am clear. Okay, coming to the chicken wire appearance. In blood vessels, oligodendroglioma and mucinous colloid carcinoma of the breast. Here the breast. Oligodendroglioma and mucinous colloid carcinoma of the breast. Fibrosis, alcohol liver disease shows the fibrosis of chicken wire fibrosis. Calcification, chicken wire calcification is seen in chondroblastoma. Now we will learn about the medulloblastoma. Medulloblastoma, it will arise from the cerebellum always. See, medulloblastoma, it will arise from cerebellum always. Coming to the genetic portion, WNT mutation is seen in the first type. SHH and NMIC mutation is seen in second type. NMIC and isochromosome 17q third type. Isochromosome 17q in the fourth type. Okay, let's revise one more time the genetics in the reverse manner. Type 4 isochromosome 17q. Type 3 isochromosome 17q and NMIC. Type 2 NMIC and SHH. Type 1 only WNT mutation. Hope I am clear. Okay. This medulloblastoma is most malignant brain tumor in children. Most malignant brain tumor in children. It spreads to CSF, shows drop metastasis. Medulloblastoma, drop metastasis, malignancy in children. Next, microscopically, it has small dense blue cells tightly packed. Blue cells densely packed with Homer-right pseudorosettes. Homer-right pseudorosette, densely packed blue cells. Densely packed blue cells and Homer-right pseudorosettes are seen in medulloblastoma. Now, this Homer-right pseudorosettes are seen in three types of tumors. That is medulloblastoma, Ewing sarcoma, neuroblastoma. All these three tumors, that is MEN tumors, medulloblastoma, Ewing sarcoma, neuroblastoma, are primitive neuroectodermal. This is ectodermal tumor. I hope I am clear about the medulloblastoma. Let's revise again. Genetics. Type 1 WNT, Type 2 SHH and NMIC. Type 1 WNT, Type 2 SHH and NMIC. Type 3 NMIC and I17q. Type 4 I17q. Next, in the cerebellum always. In children it shows malignancy, like it is actually malignant. Blastoma refers to grade 4 tumor, right? Blastoma is grade 4. In CSF, drop metastasis, densely packed blue cells, Homer-right pseudorosettes, neuroectodermal, medulloblastoma, neuroblastoma, Ewing sarcoma. This is all about medulloblastoma. Now coming to the meningioma. Meningioma, it arises from arachnoid matter, NF2 mutation. NF2 gene is present on chromosome 22, we already discussed. Next, meningioma in females, it occurs. In pregnancy, it increases in size, but after delivery, it decreases in size. This peculiar phenomenon is seen in the meningioma because it is positive for progesterone receptor. Repeating again, meningioma positive for progesterone receptor, hence it increases in pregnancy and decreases after delivery. Radiologically, dural tail sign is seen. Tumor cells are present in whorling manner, like in the whorls. I am drawing here in the green pin. Whorling of the tumor cells, whorls of the tumor cells, and summoma bodies. The calcium will also be present in the round summoma bodies. It can be of any grade. Meningioma can be of any grade from grade 1 to 4. Clinical features vary depending upon the location of the meningioma, because meningioma can occur anywhere at any location of the arachnoid matter. Hence, so we completed meningioma and medulloblastoma. Okay, now let us learn about the schwannoma. It can also be called as multiple inherited schwannoma. It is due to NF2 mutation. And coming to NF2 mutation, there are uh, three types of brain tumors in which NF2 mutation can be seen. One is schwannoma. Now we are going to study that. We already studied ependymoma and meningioma. The mnemonic is Miss Me. Here, E for ependymoma, M for meningioma. Remember, M for meningioma. We should not confuse with medulloblastoma and meningioma. Meningioma is positive for NF2 mutation. Ependymoma, meningioma, and multiple inherited schwannoma. Okay, so these are NF2 mutation positive tumors. Let us discuss schwannoma. Schwannoma, it arises from the peripheral nerve sheath because Schwann cells form the myelin sheath for neurons, right? So peripheral nerve sheath tumor. Peripheral nerve sheath tumor is schwannoma. Peripheral nerve sheath tumor, schwannoma. Most commonly, eighth nerve, that is vestibulo-auditory nerve, vestibulocochlear nerve is involved. Second most common is the fifth cranial nerve. So remember, peripheral nerve sheath tumor, schwannoma. Eighth nerve is commonly involved. Fifth nerve is second commonly involved. Microscopy is very important for Schwann cells. There are two types of zones in Schwann cell tumor. Here you can visualize one type of area, cells are very tightly packed. In one type of area, the cells are absent. This area, the cells are absent. The cells are deprived or absent in Antony B area, but cells are densely packed. Especially you can notice they are packed in two rows. This is called palisading of two rows of tumor cells. And you can also observe a pink color mass or stroma which is present between two rows of palisading tumor cells. This is called Verroque body. Vero. Vero. Verroque body. Verroque body, palisading of the tumor cells, and this zone is called as Antony A area. Cells are present in Antony A, cells absent in Antony B. Antony A, Antony B, cellular, acellular. Verroque body in Antony A. These cells, all these cells are positive for S100. Here I am rounding up the S100 positive cells. Cells are S100 positive. Okay. Now we all discussed about this Antony A, Antony B, Verroque body, and S100 positive cells. These mutations. Okay, now let us discuss the neurofibroma. Neurofibroma is nothing but a mixed Schwann cell tumors. Previously we saw in schwannoma only Schwann cells were arranged, but in neurofibroma it has Schwann cells, mast cell, fibroblast, spindle cells. Let us repeat: Schwann cell, mast cell, fibroblast, spindle cells. We all know that neurofibroma has a spindle cell, particularly CD34 positive spindle cells are present. Repeat again: neurofibroma has CD34 positive spindle cells. Mast cell, fibroblast, spindle cells, that too CD34 positive spindle cells.